According to Merriam-Webster Dictionary, evolution is defined as the process by which different kinds of living organisms are thought to have developed and diversified from earlier forms during the history of Earth. Today we are going to review all of the important topics that go into evolution, including, but not limited to, the origin of life, characteristics of life, evidence of evolution, natural selection, phylogenetics, and speciation.
Operin and Haldane's predicted early Earth conditions included a reducing atmosphere with hydrogen, water, methane, and ammonia. You might ask, how did life come about with such minimal resources that can't sustain life? Well, the Miller and Urey experiment explains everything. Miller and Urey duplicated early Earth conditions in a lab and were able to form monomers, which showed that Operin and Haldane were correct. After monomers were formed, polymers formed, protobions formed, and eventually organisms with genetic information were formed. The RNA hypothesis states that RNA was the first genetic molecule because it can easily be created under abiogenesis conditions, it can self-replicate, and it can catalyze reactions. Earth's atmosphere finally became enriched with oxygen when cyanobacteria created oxygen by photosynthesis. Some characteristics of life include responsiveness to the environment, growth, reproducing, homeostasis, being made of cells, and passing traits on to offspring. Some evidence of evolution can be found from anatomy, molecular biology, biogeography, fossils, and direct observation. For example, a homologous structure is a bone or organ that appears in different animals. So, it's an underlying anatomical commonality that demonstrates descent from a common ancestor. So when very many different animals have bones that appear to be similar in form and function, that means they can be related. Now on to one of the most important topics when it comes to evolution, natural selection. Natural selection is one of the most basic mechanisms of evolution, along with mutation, migration, and genetic drift. Charles Darwin coined the term natural selection, and he basically proved that acquired traits aren't inherited and that the traits that are passed down allow for better fitness. Fitness is a term used to describe how good a particular genotype is at leaving offspring in the next generation relative to how good other genotypes are at it. Darwin's descent with modification states that each species must have descended from a parent to an offspring, but have changed slightly over generations. So, in essence, natural selection is a mechanism by which populations adapt and evolve. Individual organisms who happen to be best suited to an environment survive and reproduce most successfully, producing many similarly well-adapted descendants. After numerous breeding cycles, the better adapted dominate. Nature has filtered out poorly suited individuals and the population has evolved. In order for natural selection to occur, there must be reproduction, heredity, variation in fitness, and variation in members of the population. Darwin proved his theory of natural selection with the study of the Galapagos finches. He observed the finches' beaks and noticed that they had adapted to the type of food they ate in order to fill different niches on the island. The difference in beak shape, despite everything else about the finches being practically the same, helped Darwin put together his new evolutionary ideas and work against the pre-existing beliefs of evolution.
In this section of the video, we are going to talk about speciation, and speciation by definition is the evolutionary process by which new species arise. There are 4 types of speciation: allopatric, sympatric, peripatric, and parapatric. Allopatric speciation occurs when there is a geographic barrier separating the two species. It literally means that the two groups live in different lands. This barrier leads to reproductive isolation and inhibits gene flow. Sympatric speciation is speciation that occurs between groups living in the same land. This is most commonly seen in plants with polyploidy and genetic polymorphism. Peripatric speciation is similar to allopatric speciation. It occurs when part of a species migrates to a certain geographic location that is different from the original. The group that moved then develops certain characteristics that distinguish it from previous species. For example, in general, there are only 2 kinds of elephants. However, in forests, you can find a 3rd type. These elephants, known as the pygmy elephants or forest elephants, have shared DNA from both African and Indian elephants. Essentially, peripatric speciation is when there is a small group that develops new characteristics and is ultimately unable to breed with the original due to its change to an isolated niche. Parapatric speciation is nearly the same as peripatric, with the one exception being the new niche is adjacent to the old niche rather than completely isolated. There are three main ways to induce speciation: geographic barriers, prezygotic barriers, and postzygotic barriers. For geographic barriers, this essentially is separating a species into two groups geographically. This includes moving one species to a new island. An example of this would be the finches in the Galapagos that were blown to another island due to wind patterns changing, and eventually those finches became unable to reproduce with the original species. This can also be called the founder's effect. Rivers, mountains, and drifting continents are also considered geographic barriers. There are 5 main prezygotic barriers that contribute to speciation: habitat isolation, which is where the habitats of the species are different so they never meet to reproduce; behavioral isolation, there's different mating behaviors; gametic isolation, the gametes of one species won't fertilize that of another; temporal isolation, mating season is at different times of the year; and finally, mechanical isolation, the reproductive organs of each species do not fit together. And lastly, we have the 3 postzygotic barriers: hybrid inviability, which is where the hybrid dies early on; hybrid sterility, where the hybrid is unable to reproduce; and hybrid breakdown, where the hybrid can reproduce, but its offspring is unable to compete with the original species and ends up dying out. These all lead to reproductive isolation. This means that the species that was once continuous can no longer reproduce and therefore begins to split off into 2 groups. With reproductive isolation comes lack of gene flow. Without gene flow, the species can eventually become unable to breed, and therefore 2 distinct species are created. So now, after speciation has occurred, we get into phylogenetics. Phylogenetics is the study of the evolutionary history or family tree of an organism, essentially who is related to whom. The relationship between species are displayed through phylogenetic trees. There are many different types of phylogenetic trees, but the one that's most common and the one that we'll be seeing today is the cladogram. Cladograms and other phylogenetic trees show the evolutionary history of an organism. This is determined by different characteristics. Today we'll focus on morphological and molecular characteristics that help us decipher the history of a species or organism. Morphological refers to the structures of the organism. You basically pick one trait and trace it back through time to see how it's changed. Where changes occur indicate where the organism has branched off or has adapted, and also where speciation has occurred. Molecular characteristics look at the DNA. You're looking at the genetic code of each organism and comparing similarities and differences. The more similar they are genetically, the closer they will be on the phylogenetic tree. Phylogenetic trees also support Darwin's view of common descent. Because they are all on the same tree, we can see that they do indeed come from a common ancestor. And that concludes our video for today. Thanks for watching.